the DNA crashes out and you can see it. It becomes this milky, goopy cloud. And this process of changing the solubility of DNA between water and ethanol by adding a little bit of salt to remove the DNA cloud that usually surrounds the DNA molecule in a water solution is how most DNA and RNA is extracted. So there's lots of protocols of how to extract DNA. One of them you can do here. I recommend doing it because it's fun and you learn something. This is the DNA extraction lab. If you have done it already in a different class, I'm sorry. Tell me about that because I don't know what other classes teach. So here you guys can see actually that the sort of cloud of DNA has been spooled here with this wooden skewer to get it out. This is vast amounts of DNA that he has in his hands right here. Genomic DNA. And then if you guys have already done the lab, another thing you can do is there's additional video on DNA extraction. In fact, there's many. On JOVE, which is Journal of Visualized Experiments. You have to be on some sort of university network to access JOVE because it's a subscription journal. So universities usually have subscriptions. Normal mortals usually don't. And I don't want you guys to pay, $30 to look at a video of DNA extraction. But what I hope you carry away from these two slides is that DNA and RNA chemical properties, the fact that they are negatively charged, facilitates both the solubility of DNA in water and the extraction of DNA in ethanol. Now, another thing that you can use the negatively charged property of DNA for is for separation. So you can take differently sized DNA and RNA, never mind why they're differently sized for now, and you can easily separate them by size when you put them into this gel-like environment. So a gel is, basically gel is just a fancier Jello with less impurities in there. Gel is a partially solid, partially liquid sort of state, which is saturated in water and has lots of holes. But those holes are sufficiently small that a large DNA molecule will get slowed down, as you can see in this picture. The large blue molecules are moving slower than the really small green molecules, and the little small green molecules move faster than red molecules that are a little bit smaller than blue, but a little bit larger than green. So only because DNA is negatively charged, and RNA as well, can you put the DNA into an electric field with a negative electrode where you're loading the DNA, and the negatively charged DNA will move towards the positive electrode, and the smaller sized DNA will move faster, and the larger sized DNA will move slower. And this will allow you to actually look at different DNA subspecies that you have created in your genomic DNA by, say, doing restriction digest, which we'll talk about in some other day. And also, so just like you can do a little lab about DNA extraction, you can do a little lab about gel electrophoresis, because these two techniques are very basic molecular biology techniques. If you guys are getting a degree in molecular biology, you should know them by heart, and I hope that in your actual experimental labs, I'm sure you're going to get to do them. And this picture just shows you how the total genomic DNA will look on an agarose gel. So people are very used to DNA looking like little bands. Normal DNA, unless it has been broken or cut and sheared in some way, will never look like a little band. Genomic DNA is just a really large molecular weight smear here, and you can see that there's a little bit of smearing towards the end, towards some more faster moving species. And when you see that there's basically no bands at all in here, that just means that some of the really large molecular weight genomic DNA broke down during the extraction process, and now they're randomly sized fragments of DNA because there's so many of them that they of all kinds of different lengths, you get a smear. It's usually a sign, the more smearing you get in your DNA gel, if it's just genomic DNA, it's suggestive of some sort of degradation that has occurred. Your DNA somehow broke. So, and then one thing, this slide I just skipped, is to talk about the way you can make DNA and RNA visible on a gel electrophoresis experiment. Because the DNA by themselves, yes, it absorbs ultraviolet light at 260 and 280, but it's hard to see absorption on a gel. What is easy to see is fluorescence. What fluorescence is when the ultraviolet light hits a particular molecule that can fluoresce, the molecule will then release light back of a different wavelength. In this case, ethidium bromide will absorb blue ultraviolet light and then release an orange colored light of a higher wavelength. Because you have a lot of blue in the background, they usually look pink on the gels, but they're actually orange. And so this dye called ethidium bromide, not only is it a fluorescent molecule, there's a lot of fluorescent molecules out there, a bunch of them having these benzene rings, which are good for fluorescence. It also is able, because it's just the right size, to intercalate or squeeze itself between the base pairs of the DNA molecule. It squeezes itself into the DNA and then it glows, and that's how you see the bands on the gel. But what you can see here, because ethidium bromide intercalates itself into the base pairs, it changes the DNA structure. There's no longer quite as need of a base stacking. There's some, because ethidium bromide is in the way, the DNA molecule becomes less dense. It's considered, because it alters the DNA structure, it's considered a mutagen. So whenever you work with ethidium bromide, you have to wear gloves. It's a really large molecule, so it should not be going through your skin barrier, but if you have a cut, well, then it can get into your bloodstream and then potentially wreak some havoc. And I'm going to take a break right now. Hi. So one more concept I'd like to cover for the DNA structure part is the chemical DNA synthesis, which is explained at the end of the chapter 6. I think it's a very important concept, and it's a little bit more chemically involved than the gel electrophoresis and DNA extraction we covered earlier for this topic. Now, you guys don't need to remember the actual chemicals involved in the process, but hopefully you will appreciate a few finer points about DNA synthesis. First of all, if the DNA is synthesized chemically, as you can see on this diagram from the book, it's actually synthesized not the way DNA polymerases do it. It's synthesized not 5' to 3', but 3' to 5'. That's the chemical DNA synthesis way. So let's start with the basic idea. You can see here that the process starts at number 1, where the first nucleotide of the molecule to be synthesized has been attached to a support bead for ease of isolation later. The first nucleotide has been added, so it has some sort of base, whether it's A, C, T, or G, and the ribose. And then instead of the phosphate group, it has this DMT group here, which is a protecting group. So step 1 is to remove the DMT group and allow for the actual chemical reaction of attaching the next nucleotide to occur. Then the next nucleotide is flown into the synthesizer, activating, and this nucleotide has an activating group, which is this complicated chemical structure here, called diisopropamylamino activating group. You don't have to remember that. When the group reacts, the byproduct is released, and now a linkage is formed between the first base, which is the blue one, and the second base, which is the yellow one. And it's not a phosphodiester linkage yet. It's a phosphite linkage. Now, an oxidation occurs in the synthesis chamber, and now the linkage has been changed to a phosphodiester linkage. However, there is still a second protecting group which will protect this phosphodiester from cross-reacting with the subsequent nucleotides that are flown in, that are put into the reaction chamber. And the process will repeat itself. Now, the DMT of the second base is going to go through the removal process. A third base will be flown in, a phosphate linkage is formed, an oxidation occurs, and a third nucleotide has been added to the growing DNA chain, and it is added into the 3' to 5' direction. So it's opposite from what DNA polymerases do. Then once depending on the lengths of the DNA to be synthesized, once all of the nucleotides have been added, then the protecting groups are removed from the bases. And these cyanoethyl groups that have been covering the phosphates and preventing them from cross-reacting are removed as well. And the bead is detached. The primers or the oligonucleotide chain is removed from the support. So you guys can see that this is a single-strand DNA molecule that's being synthesized, not a double-strand one. And then this picture doesn't show it, but then all primers will be checked on by size exclusion. So they will the company will run them on not on a gel usually, but on a chromatography column to exclude everything that's shorter than than the primer they were planning to synthesize. So if you're trying to synthesize a DNA stretch that's say 20 base pairs long, you will exclude everything that's less than 20 because all of those stretches will have some sort of misincorporation of a nucleotide. So all primer or DNA synthesis companies perform this cleanup step. And of course, the longer the DNA molecule that you're trying to synthesize, the more likely a mistake will occur. So if the company is synthesizing something like 100 base pairs, they will have further purification methods to assure that this was the only thing that they then send you in a tube is the DNA primer of that particular length, and they are not introducing some sort of mutations into the primer that they're sending to you. This is just a short list of the typical chemical DNA synthesis companies. IDT, or Integrated DNA Technologies, is probably the most frequently used. Eurofins or Operon is another company. And then there's now companies that will synthesize much, much longer DNA fragments because they use a combination of DNA synthesis and gene synthesis, where they overlap a bunch of DNA stretches to make a much bigger stretch. So GeneScript is one of them, IDT does it too, and so forth. And now this is just a summary slide for the whole DNA structure lecture. I just want for you guys to take away the most important points of what I was talking about. So DNA is a repository of biological information, as you guys probably all know. And earlier during this lecture, we talked about how that was found. So the experiments of Griffith, the experiments of Avery, and then the experiments of Franklin, Watson, and Crick. The important parts about the structural aspect of DNA is that both DNA and RNA molecules have directionality. They're 5' to 3', that the DNA or RNA helix is always anti-parallel, that you can break the DNA helix by either putting into a polar environment such an acidic or basic environment, or heating it to break up the hydrogen bonds between the bases, which is the thing that's hold the double helix together. That the DNA and RNA molecules are negatively charged, and it has been extensively used for working with DNA. So DNA is extracted because the solubility of the DNA can be changed in different solvents. So it changes between water and ethanol. And you can separate DNA by size. If you put them into an electric field with a negatively charged DNA, will travel towards the positively charged pole. And because it's in a gel environment, which is not liquid but gel-like, so there's holes that the molecules have to squeeze through. The DNA can be separated by size. That the DNA can be made visible by fluorescent dyes that bind DNA, for example, ethidium bromide, but there's plenty of other dyes that do that as well. And that DNA or RNA can be synthesized chemically as well. So hopefully this was a helpful lecture, and I see you later. Hi. So I wanted to address an important point from the DNA structure video, and that is DNA directionality. The reason I want to talk about it is because I think it's easy for me to talk about it using a blackboard. And what I want to talk about is how you design a primer that matches a particular DNA segment. To remind you guys, a primer is just a short stretch of DNA. The word primer originates from the idea that DNA polymerases can start synthesizing a strand of DNA de novo, just from scratch. And they need either a primase that will make an RNA primer, or an end of a DNA molecule in order to start attaching nucleotides. When various DNA technologies are used, the name for that short oligonucleotide is a primer. So let's say there's a stretch of DNA and because I hope you remember that whenever we write a DNA molecule, its orientation is implied to be 5' to 3'. So there's a phosphate here and there's an OH here. And let's say I'm trying to design or figure out if a particular other molecule will bind this DNA fragment. And I only wrote a few nucleotides, but let's pretend there's, I don't know, 30-something or 100-something nucleotides. So, because we hopefully all know from back from biology class that A's pair up to T's and G's pair up to C's, it seems trivial to write down the molecule with complements, well, base pair versus DNA molecule, right? You just match this up, A's match to T's, C's match to G's, and so forth. But because DNA helixes and RNA helixes are always in an anti-parallel orientation, the orientation of the second DNA strand is now 3' to 5'. So it's actually written incorrectly. It's written in reverse. And if you were to actually write this DNA molecule for purpose of communicating what this DNA molecule is going to look like, whatever this DNA molecule is, oh my gosh, messed up. You would be writing it in reverse, okay? So you would always start with the 5', right there. And then just match the bases. So C comes first, then T, then T. Like that. And that's super important because both in your quizzes and your exams we will test you on this knowledge and your ability to figure out which of the nucleotide stretches match up with the template, which ones of the nucleotide stretches will base pair, because it's key knowledge for your ability to then design PCR primers, cloning, and all kinds of molecular biology applications. So, this is how you need to write it, all right? And this is, I'm gonna stop.